Okay, for those who have joined us, we're going to be starting in just a couple of minutes. We're going to wait for more of our participants to join. So we'll be starting this webinar in just a couple of minutes. Just going to wait a little longer for more of our attendees to join, and then we will get started with this webinar. Okay, hi everyone. I think we're going to get some more attendees in the next couple of minutes, but I might as well start the introduction. My name's Hannah Jones, and thank you for joining the third MASTS webinar that is happening in our series. Today's speaker is Irma Cascao from the University of the Azores, and before I hand over to her, I'm just going to do the generic housekeeping about the webinar. So after Irma's presentation, we will be doing a live Q&A. If you would like to ask a question, please use the Q&A box that is at the bottom of your screen. You can type that question in at any point, and we will work through those questions after the presentation. You can also ask questions anonymously if you wish. This webinar is scheduled to end around 1:30 UK time, but I've got the thumbs up from Irma that we're okay to go over that time if we have questions that takes us beyond that 1:30 end time. This session is getting recorded as well, so if you missed something, want to recap, send it to a colleague, or maybe you're not able to stay for the entire session, you will be able to find the recording on the MASTS YouTube channel, along with all our previous videos that have included previous webinar series as well. So, our presenter. A quick introduction to Irma. She's originally from South Africa and did her marine biology undergrad in Lisbon, and in her final year began to work on projects such as photo IDs of bottlenose dolphins. And in 2005, she undertook a 3-month internship in the Azores with the Azores Whale Lab and the university, and has been participating in several research projects specializing in hydroacoustics, passive acoustic monitoring, and cetacean bioacoustics. In 2018, she finished her PhD with the University of Azores, which looked at the impact of seamounts on cetacean-prey interactions by using passive and active acoustic techniques. And since her PhD, she is still at that university, and she has been recently working on various projects, including studying the impact of underwater noise on marine ecosystems. The work that Irma is talking about today was funded by the Saltire Emergings Researcher Scheme, which was a joint collaboration between MASTS and the Scottish Funding Council, and it allowed Irma to come to the University of St Andrews for 7 weeks and to work with Andrew Brierley's lab at the University of St Andrews. So that is enough from me. I'm going to hand over to Irma, so hopefully she can start sharing her presentation and turn her camera on. And that's it from me, so over to Irma.
Thank you, Hannah, for that introduction. Hi, everybody. So as Hannah said, I'm a postdoc researcher at the Institute of Marine Science, Oceans in the Azores. So first, I would like to thank the MAST Exchange Programme for supporting my visit at the Scottish Ocean Institute in St Andrews, and to have had this opportunity to work closely with experts in this research field. This exchange is part of an existing research collaboration between our lab, Azores Whale Lab Cetacean Ecology Group, and the Pelagic Ecology Research Group, in the scope of two ongoing European projects, SUMMER and MEESO. The ocean mesopelagic zone, so approximately between 200 and 1,000 meters deep, harbors highly diverse, abundant communities of fish, cephalopods, crustaceans, and jelly-like animals. So these organisms are abundant worldwide and form a large biomass in oceanic waters. So most mesopelagic animals undergo diel vertical migration, so rising towards the surface at night to feed and returning to deeper waters during the day. This represents the largest movement of biomass on Earth. So therefore, these organisms are believed to play a major role in the transport of carbon and nutrients between mesopelagic and epipelagic environments. They are critical components of oceanic food webs. So they consume large quantities of phytoplankton and small zooplankton and serve as primary prey for many top predators, such as demersal and large pelagic fishes, squids, seabirds, and marine mammals. Yet, there remains considerable uncertainty about the biomass and trophic importance of mesopelagic, because we lack fundamental information on species composition, biomass, spatial distribution, and vertical migration. The main goal of this exchange is to contribute to Project SUMMER and MEESO that aims to fill in these and other gaps in knowledge about mesopelagic organisms. More specifically, to determine the best combination of methods to obtain accurate estimates of the biomass and biodiversity of the mesopelagic community, and to provide regional and global estimates of mesopelagic fish. Therefore, the purpose of the exchange was to improve skills in analysis of acoustic data collected from scientific echosounders off the Azores, to apply pre-processing methods to remove noise and improve data quality, estimate acoustic density of mesopelagic organisms, characterize their diel vertical migration patterns, and finally, estimate the contribution of fish to the overall acoustic backscatter. The Azores is the most remote oceanic archipelago in the North Atlantic Ocean. It is located at a triple tectonic plate junction, and the mid-Atlantic ridge runs across the archipelago. The Azores consist of 9 volcanic islands, shown in green, separated by deep waters with scattered seamounts. So seamounts are common topography features in the Azores economic exclusive zone. In the study, the acoustic data were collected using Simrad EK500 scientific echosounder operating at 38 and 120 kilohertz aboard the research vessel Archipelago, and using the Simrad EK80 scientific echosounder operating at 4 different frequencies aboard the research vessel Ocean Explorer. So an echosounder system contains a transmitter that generates bursts of electrical energy. So a transducer mounted on the hull of the vessel converts the electrical energy into a sound pulse, a ping, at discrete frequency that propagates through the water. So any objects that have a different density from that of the water, for example, a fish school, will reflect the transmitted sound waves and generate backscattering waves, echoes, which are detected by the transducer, converted back to an electrical voltage, and displayed graphically as an echogram. So the echo intensity scattered by the organisms is given in terms of volume backscattering strength. Since most acoustic scatters were aggregated in schools and layers, single target echoes could not be identified. So therefore, acoustic density of organisms was estimated by calculating the mean volume backscattering strength. Here are examples of echograms showing diel differences between regions. So warmer colors indicate a higher volume backscattering strength. You can see the bottom right echogram shows the upward migration of the deep scattering layer in open waters at night. So acoustic surveys aboard the research vessel Archipelago were conducted at 6 locations. So 4 seamounts in open waters and in the surrounding area south of Fayal Pico Islands. So all surveys followed a predefined transect. Day and night samplings was equally distributed at each study location, but most of the survey effort was done in summer due to good weather conditions. So far, only 38 acoustic data collected at Condor and Gigant and in open waters have been already analyzed during my PhD. In June 2021, a joint mission with OceanX was conducted to explore the foraging, ecology, and acoustic communication of sperm whales. So while tracking sperm whales for tagging, concurrent acoustic data and water samples were collected to characterize the prey and oceanographic landscape to contextualize sperm whale movements and behavior. Here, all acoustic transects followed an adaptive sampling approach. The obtained water samples were sent to ATSI, not sure how to say that, partner of SUMMER project for eDNA analysis. So the ongoing analysis involves the analysis of the acoustic data collected concurrently with eDNA to identify species composition and compare estimates from both methods, and the analysis of the large acoustic data set collected of the Azores to estimate the distribution and abundance of mesopelagic organisms in the region, based on the biodiversity information acquired above. Unfortunately, I'm still analyzing the data. I do not have results yet to show, but I will in this presentation show some results obtained during my PhD related to this topic. But first, I would like to highlight that the main outcome of this exchange is the improved skills and increased knowledge in processing and analyzing analysis of acoustic data, particularly in using EchoView software. So for instance, the application of pre-processing methods to remove noise and improve data quality, and the multi-frequency techniques to discriminate between taxonomic and size groups. And plus, I gained new skills in using Python programming language to develop a custom algorithms for processing acoustic data. So the expected outcomes will be to establish a protocol for density estimation of the mesopelagic organisms. So to ensure standardization of procedure, as well as the development of an efficient school scattering layer detection in Python created by Dr. Roland, to classify and characterize the mesopelagic community. So both outputs are critical tools for the analysis of echosounder data, and well beyond this collaboration and the projects. And most importantly, the analysis of our acoustic data from the Azores will contribute to SUMMER and MEESO deliverables in the provision of abundance estimate and biodiversity information, so therefore contributing to increased knowledge and preserving biodiversity and ecological functioning of mesopelagic systems, and ensuring that any future harvesting is done sustainably without compromising the critical ecosystem service provided by these groups, for example, carbon sequestration and the role in the trophic webs. So now I'm going just to present you some results obtained during my PhD at 2 seamounts, Condor and Gigant, and in surrounding open waters. So over the summit, acoustic density was 18 times higher than over the slopes and 10 times higher than in open waters. The lowest backscattering occurred above the slopes. Estimates of acoustic density were similar between Condor and Gigant. In open waters, the highest backscattering occurred during the day, while the reverse pattern was observed in Gigant. Diel difference in Condor were small. In Condor, there was a clear seasonal pattern with an obvious peak in spring and decreased greatly in summer and autumn. Whereas in Gigant and in open waters, variations were not so clear, with a slight increase during the summer. So maps of predicted spatial density obtained from the best game model were produced to visualize the distribution of acoustic scatters at unsampled locations of the study areas. So warmer colors indicate higher acoustic densities. So you can see that the highest mean backscatter was constantly found over the summit. So regardless of the season and diel period. And the acoustic density decreased drastically along the slopes, reaching the minimum values at the base of both seamounts. And a slight increase was predicted towards deeper waters away from seamounts and in open waters. So seasonal patterns were more, you can see here, seasonal patterns were more pronounced in Condor, with much lower backscatter in autumn over the slopes and in areas away from the seamount. While diel patterns were much more pronounced in Gigant, with constantly higher nighttime values over summits and slopes. This is a graphic view of the vertical distribution and spatial structure of the sound scattering layers at seamounts and in surrounding open waters. So the sound scattering layers were permanent features with 2 main layers. So we had the shallow scattering layer and the deep scattering layer. The vertical extent of the colored rectangles indicates the vertical distribution range of the layers. The solid horizontal line within the rectangles indicates the mean depth of the scattering layer. Warmer colors indicate higher acoustic densities. And the fish drawings represent the amount of spatial structurings, strong, moderate, or weak of the scattering layers. So that is, closer fish indicates strongly structured layers, while a dispersed fish indicates weakly structured layers. So our findings suggest the following. So first, over the summits, a community of non or weakly migratory fish live a mesopelagic lifestyle. So remaining close to the seafloor during the day and dispersing to feed at night. Deep scattering layers found over slopes and open waters migrated towards the surface at dusk to feed, but an intense deep scattering layer persisted overnight in deep waters, indicating presence of non-migratory organisms or migrants from depths greater than 1,000 meters. Fishes may be advected onto the summit and become entrained in the circular flow generated by the tailor cap. Or fishes may actively seek seamounts to take advantage of the prey aggregated around the top by the tailor cap or by the seamounts topography. Finally, as the migratory fish starts descending towards deeper waters the next dawn, fishes above the summit may become trapped and aggregated close to the bottom of the summit and upper slopes. So to conclude, persistence of high densities of micronectin aggregated over seamount summits, regardless of the season and diel period. Such high densities may be explained by the presence of seamount-associated micronectin community and/or by the retention of vertical migrating micronectin. More information about this work can be found in our papers, and Hannah will be sharing you the link if you're interested. So thank you all for listening. And again, I would like to thank the MAST Exchange Program for the funding and for providing the opportunity to meet and work with Professor Andrew and Dr. Roland. Thank you.
Great, thank you so much, Irma, for that talk. If anyone wants to drop Irma a line, her email was just on the screen there, and I'm sure she can drop it in the chat at any point as well. So if you have a more in-depth question, please do drop her an email. But also, we have her on the line right now, so please do use the Q&A that is at the bottom of the screen if you wish to ask a question to her now. I have a couple of questions myself to kind of kick off the kind of conversation. I'm sorry if you already said this, but I actually don't know what the term micronectin is. What is that?
So it's dense aggregation of zooplankton and micronectin
so is it like a community that are found in certain parts of water?
Yeah, it's a mixture of, like I said, fish, cephalopods, and crustaceans and jelly-like animals. So they create these dense aggregations. And micronectin is they're small animals, so between 2 and 20 centimeters. So,
okay.
Yes.
Right, okay, because yeah, I didn't know what that phrase was, and I was like, what is this? So one of the things I wanted to also ask you is, do you have any other field work that is this kind of work happening elsewhere, not just in the Azores where you're located? Do you know of are you planning on doing anything else anywhere, or know of work that's happening like this?
So you're asking about me, right?
yeah. If you have any other field work coming up, is it always focused on the Azores, or do you plan on taking it anywhere else?
Yeah, it's been always focused on it's been always focused on Azores. So we've done the transects in the past, and then after unfortunately our echosounder broke down. And finally, due to the collaboration with MEESO project, they borrowed their echosounder. So now we're starting again slowly to do more transects and more to collect more data. So, but yes, it's been here in the Azores, everything. So yes.
Okay, that's great. I was just curious about that. Andrew Briley very kindly put into the Q&A that micronectin are crudely animals that are bigger than zooplankton but smaller than fish. So yeah, it's quite a broad term. I get that now. We have another question in here as well, saying as species that they are widely distributed, how do you study the distribution of the species? I do find it hard to delimitate a distribution of species highly moved. I think they're saying that the distribution of species that move a lot, especially in a small relative area. Is that a question that you can kind of dissect, or do we need a bit more information?
Of the species.
Gemma, if you're able to maybe provide more information about which species you're referring to, or is it just the community? They mean mesopelagic fish that move a lot.
Yes. So we so as I said in our talks, we use the echosounder and we use the transects to be able to study these animals. So of course, each location, each place. So lately we've been focusing our work on seamounts, and yeah, I'm not so sure if I, sorry.
That's all right. Gemma, if you want to maybe drop Irma a line, maybe you can just provide a bit more explanation to your question, and hopefully Irma can give you the information that you need. So we have a question here from Hayley saying, thanks for the talk. Have you started looking into the links between acoustic backscattering and sperm whale distribution? If so, what are the links you are expecting to see?
Yes, we had this mission in 2021 that we tried to tag the sperm whale at the same time we were doing the acoustic surveys. Unfortunately, the tagging didn't go so well, because we were trying with a new system that the tag had this camera, so we can try to also understand. But during that survey, we collected water samples. So in the area of where the animals were foraging, we would collect water samples. In areas, non-foraging areas, we'd collect. So we're going to try to use the acoustic data and the eDNA analysis and try to see if we can understand, you know, the link, and if we can get anything. But unfortunately, the tag data with the sperm whales would be perfect. It's not been easy to do. So.
Okay, so a question from me. You mentioned working on the Python kind of code. Is this something that will be open source, that you know, other research groups will be able to look at themselves?
So Python is a completely new thing for me, which thank you for Roland to introduce me into this world, because I'm used to with R or MATLAB, so he has created this scattering layer detection algorithm, and we're going to try to see, we're going to work together to develop an efficient algorithm, and hopefully, I think, yeah, the object, the main purpose is an object we can share and will be open. So, but yeah, hopefully.
Okay, so work in progress right now.
Yes.
If anyone else on the line has a question for Irma, please do use that Q&A box at the bottom of the screen, and we can run through those questions. The projects that you mentioned you're contributing to, SUMMER and MEESO, are they just European focused, or are there contributors to those projects all around the world? Like, what's the kind of basis for those?
It's European. Yeah, it's just European. Yes.
Okay, great. And we have a question here from Pablo saying, what organisms inhabit the summit seafloors in your study area? Could the high biomass there be related to it?
Yeah, the thing with the acoustic data is that we don't know what type of species. Okay, so maybe now with this, because I only analyze the 38 kilohertz, and I learned now with this MASTS program with Roland and Andrew, so we can try to use multi-frequency to try to identify what type of groups. Of course, we know we've got few data, for example, on condor seamounts, of certain fishes that they're catched with the bait camera that they deployed. So they just like saw a few myctophides. Of course, you got the demersal fishes, and you got the I think with the multi-frequency analysis, we can try to understand more what's going on.
Okay, great. So that brings us to the end of the questions that we've got so far. So if anyone here Yeah, I want to ask a question, please type it in the next minute. Remember, this recording is going to be uploaded to the MASTS YouTube channel, so if there's anything that Irma spoke about today, you can recap it on the MASTS YouTube, or you can even send it to a colleague who may have missed this and you think that they should know about it. And it might be worth maybe you dropping your email into the chat for the audience if they want to follow up with anything as well. We do have another question in here, which I'm just going to allow you to type first, and then we'll get into that question.
Oh, good. Just wait.
That's all right.
Yep.
Great. So Irma, there is a question from Richard here, which is asking, how successfully was eDNA analysis in discriminating fish and signals from schools?
So we still haven't obtained the data. So everything is still in the lab working. But you know, there has been studies that's been recently published, and it looks that it's going to be a very good tool to be able to help, especially with acoustic data, because, you know, for example, trawling is also a good way to be able to identify the species, but of course, you know, the animals escape. So it's also not very easy to, you know, to get the accurate estimate. So hopefully we'll see if this type of analysis will be a good tool or not to be able to discriminate.
And I assume with the eDNA sampling, you're taking it at various depths.
Yes, we done.
So all the way down to.
We done all the way until 1,500 meters, because you know the sperm whales, they can, at least the whales that we've tagged here, they've been diving between 1,000 1,200 meters. So we try to catch the whole water column during the day and night, so to be able to catch the whole picture, and in the foraging areas and non-foraging areas to see, you know, if there's difference or not. So I'm really excited and hopefully waiting for the results, and also when we finish analyzing the acoustic data to see what we get. So yes.
Great, thank you. So we've worked through all the questions that have been submitted, and we are now gone past the finish time as well. So I have put into the chat the links that Irma referenced, and her email is in there too. That leaves me to say thank you very much, Irma, for being today's presenter. I hope you enjoyed answering those questions, and it was a great talk and a very useful infographic, by the way. I really liked your infographic with the you know, the colored squares showing where the fish went. I think there was a lot of information that was conveyed in such a simple image. That was really great. What I've put up on the screen for everyone who's still with us is what the next MASTS webinar is all about. So all you have to do is head over to the MASTS website if this is a talk that you would find interesting. It's all about modelling, productivity and sustainability of macroalgae in the North Atlantic under various climate change scenarios. So this is in 2 weeks' time at 1 o'clock on the 29th of March. If you would like to join this, please do sign up for it and check out what all the next talks are going to be. This was our third of, I think, in total 12, maybe 15 talks. So if there's a name in here that you recognise, please do sign up for that talk. You can see all the talk titles on the website. That's the MASTS website. And we really do hope that you can join us in the future for any of our talks. So thank you again, Irma, for being today's presenter. And eventually everyone will find the recording online as well. So thank you everyone for joining, and hopefully we will see you soon.
Thank you.